Good day. My name is Nathan Standifer, and I am an Associate Director and the head of GXP New Modalities Bioanalysis for AstraZeneca PLC. Today, I'm going to discuss the use of multivariate analytical techniques to assess immunophenotyping data for associations with immuno-oncology therapeutic dose and clinical response. The first slide provides a synopsis of the key points to be provided today. I'd like to only highlight the first two bullet points in which the rationale for monitoring lymphocyte populations in IO trials is detailed. Basically, our current mechanistic understanding of IO therapeutics is that they facilitate activation of tumor-reactive T cells upon encounter with antigen. This can, of course, be monitored using validated immunophenotyping assays to detect and quantify discrete activated or proliferating T cell populations in patient blood specimens. Now at AstraZeneca, we've developed numerous flow cytometry-based immunophenotyping assays for use in clinical studies. The assays shown here represent our current core panels implemented in most IL trials. In brief, we monitor T, B, and NK cells, regulatory T cells, proliferating T cells, and naive and memory T cells or activated T cells. Monitored activation markers include HLA-DR, CD38, or ICOS. Importantly, results of these assays can be reported as both population percentages and absolute cell counts in cells per cubic millimeter of blood. In addition, we monitor relative expression levels of ICOS and the proliferation marker Ki67. And we do this by recording the molecules of equivalent soluble fluorescence using quantification beads to quantify fluorescence. Now, these core assays were bioanalytically validated based on context of use at Q-Squared Solutions, our selected flow cytometry testing vendor. As each assay has multiple reportable populations, the ranges of percent coefficients of variation for imprecision and stability are listed. Overall, all reported populations exhibit less than 20% coefficients of variation for imprecision and are stable for up to 48 hours at ambient temperature in the presence of up to 300 nanograms per mil of drug. So today I'm going to focus on two IO therapeutic molecules, both of which are under development at AstraZeneca. The first, shown on the left, is durvalumab, or known by its brand name, Imfinzi. Now, durvalumab is a modified monoclonal immunoglobulin G1 molecule with specificity for the programmed death ligand 1 molecule, also known as PDL1. Durvalumab is currently approved in 2 indications in both the United States and the European Union. Unresectable stage 3 non-small cell lung cancer as maintenance therapy following chemoradiotherapy, and also extensive stage small cell lung cancer in combination with etoposide and platinum-based therapy. Tremelimumab, shown on the right, is a monoclonal immunoglobulin G2 with specificity for the cytotoxic T lymphocyte-associated protein 4, also known as CTLA-4. Based on its isotype of immunoglobulin G2, tremelimumab does not mediate effector functions such as complement fixation or antibody-dependent cellular cytotoxicity, and thus it purely blocks interactions of CTLA-4 with CD80 or CD86. Currently, tremelimumab is not approved for marketing in at any country in the world. Today, I'll be focusing on a study known as Study 22. This is a phase 1 and 2 trial of durvalumab and tremelimumab alone or in combination with in advanced unresectable hepatocellular carcinoma patients. I'm only going to focus today on the phase 2 portion, which is shown in the right 2 ovals in this slide. And in this phase 2 portion, hepatocellular carcinoma patients, also known as HCC patients, were randomized into each of 4 treatment arms, shown most clearly in part 3. In the durvalumab monotherapy arm, patients received 1,500 milligrams of durva every 4 weeks, also known as Q4W, for 1 year. Patients receiving tremelimumab monotherapy received 750 milligrams Q4W for 7 doses, and then they received a Q12W regimen. There were 2 combination therapy arms. In the first, termed T75 plus D, patients received 75 milligrams of Tremi Q4W for 4 doses, plus durva at 1,500 milligrams Q4W for the trial duration. In the second combination arm, termed T300 plus D, patients received a single dose of tremi at 300 milligrams and durva at 1,500 milligrams Q4W for a year. The rationale behind this dosing regimen was to elicit immune cell activation and maintain this with monthly durva administration while minimizing the toxic effects of repetitive CTLA-4 blockade. In this slide, the initial Study 22 clinical results are shown as of February 2020. As you can see from the Kaplan-Meier plot, the highest median overall survival of 18.7 months was observed in patients enrolled in the T300 plus D arm. And this is shown as the purple lines and symbols in the Kaplan-Meier plot. This was followed by the tremi monotherapy arm with a median OS of 15.1 months, shown as the green lines and symbols. Interestingly, patients enrolled in these arms exhibited extended overall survival that was evident as early as 5 months compared to patients receiving durva monotherapy or the T75 plus D regimens in the blue and red lines and symbols shown respectively. In terms of the analytical strategy for immunophenotyping data, our goal was to assess any associations between monitored lymphocyte populations and dose. Since we collected data on 26 discrete lymphocyte populations from each enrolled patient, we opted to use quadratic discriminant analysis. QDA is a supervised categorization and dimensionality reduction algorithm that is used to derive combinations of variables that maximally discriminate among known groups. For Study 22, we constrained the algorithm to assign subjects to one of 4 groups corresponding to the number of enrollment arms. This was, of course, based on looking at day 15 lymphocyte population counts. The results of the QDA are shown in the next slide. Each patient is indicated by a circular filled symbol, the color of which corresponds to the algorithm assigned group or arm. The crosses in the figure represent the multivariate means for each arm, and small and large ovals represent the 95% confidence intervals and 50% contours, respectively. The axes represent the canons, which are the algorithm-identified combinations of variables, in this case, lymphocyte populations, that maximally delineate membership of each patient in a group. The QDA was highly accurate in that it assigned 98.3% of patients to the appropriate treatment arm with a 1.7% misclassification rate. This corresponds to 2 of 119 patients being miscategorized. Also, the entropy R² value, which is a measure of the fitness of the model, was 0.92. A perfect fit is a value of 1.0. Canon 1 in the plot represents the mix of lymphocyte populations that maximally discriminate group membership, and this is shown on the horizontal axis. Notice that Canon 1 values increase with increasing tremi dose along the horizontal axis, such that patients who receive durvalumab alone are located on the far left, and those who receive the high-dose tremelimumab are located on the far right. However, for Canon 2, which is shown in the vertical axis, only patients in the T300 plus D arm were distinguishable from other arms. So next, we wish to identify the lymphocyte populations most associated with each of these canons, and we began with Canon 1. To do this, we used linear regression to assess the correlations between day 15 values for each of the 26 lymphocyte populations and the Canon 1 score shown. We set an arbitrary correlation cutoff value of 0.1 and listed all populations with an R² value greater than 0.1. As you can see on the left side of the slide, all populations meeting this criterion were CD4 positive T cell populations, with the top most strongly associated being proliferating Ki67 positive CD4 positive T cells and ICOS positive CD4 positive T cells. Interestingly, all populations associated with Canon 1 exhibited tremi dose-dependent increases, most clearly observed in the box plots on the right of the slide. Therefore, we can say that Canon 1 associated populations are those that increase as a function of tremelimumab dose. Next, we repeated the same strategy for Canon 2 in each of the 26 populations. Recall that only patients receiving T300 plus D exhibited Canon 2 elevations. In this case, we only observed two populations meeting the acceptance criterion, and those included CD8 positive Ki67 positive cells and CD8 positive ICOS positive T cells. Analysis of the relationship between treatment arms and proliferating CD8 cells, or CD8 Ki67 T cells, revealed a saturable relationship in which maximum counts were observed in patients receiving the T300 plus D, and the counts did not increase in patients receiving twice as much TREMI, or those receiving the T monotherapy. And that's shown on the right-hand side of the slide in the box plots. Similar associations were observed with the CD8 positive ICOS positive T cells. These findings reveal that some other variable must account for these Canon 2 associations, and we next sought to investigate these potential variables. As patients receiving T300 plus D demonstrated the longest median overall survival, we sought to investigate any relationship between Canon 2 populations, that is CD8 Ki67 positive T cell quantities, and clinical response. Toward this end, we assessed day 15 proliferating CD8 T cell counts from each treatment arm, stratified by best overall response category, where CRPR in this figure stands for complete or partial response, SD stands for stable disease, and PD stands for progressive disease. As you can observe, patients with complete or partial responses demonstrated elevated proliferating CD8 T cell counts compared to patients with progressive disease across all treatment arms. In addition, the highest median CD8 positive Ki67 positive T cell counts were observed in patients receiving T300 plus D, and that's shown in the purple boxes in this figure. Thus, these findings link clinical responsiveness to combined checkpoint blockade with an acute T cell expansion. Moreover, these data demonstrate that both the quantity and quality of immune cell activation is important. In other words, even though patients receiving TREMI at 750 milligrams demonstrated the highest level of CD4 T cell activation, as shown in a previous slide, patients demonstrating the highest CD8 T cell activation levels, as shown in this slide, exhibited the best overall responses. So in summary, we've used quadratic discriminant analysis to identify two sets of lymphocyte populations that maximally discriminated IO-treated patients into appropriate treatment arms. The first was comprised of CD4 positive T cell populations, and these populations increased as a function of TREMI dose. The second was associated with positive radiographic response. So by using multivariate analysis, we were able to identify a possible mechanistic underpinning to the initial clinical observations of improved response to the T300 plus D treatment. So from the perspective of clinical flow cytometry, the use of this and various other multivariate analytical tools will become more prevalent and essential, particularly with the advent of ultra-high dimensional flow cytometers. Such means of exploring immunophenotyping data hold great promise for the selection of optimal dose, identifying correlates of clinical response, and ultimately gaining a better mechanistic understanding of IO mechanisms of action. I thank you for your time.